would like to introduce Dr. Enie Hensel. So Dr. Enie Hensel is a senior postdoctoral researcher at UF's Nature Coast Biological Station, working with Savannah Berry and Laurel Reynolds. She is an especially important person to me as one of my mentors and coworkers, so I'm so honored to introduce her to you all. Enie completed her undergraduate degree at UF and earned her PhD in ecology and evolution from NC State. Enie's work focuses on reducing human-driven stressors in coastal habitats and improving ecosystem resilience. She blends hands-on fieldwork with input from local stakeholders and is especially interested in how habitat structure and predator-prey interactions shape coastal systems. Her goal is to produce science that supports conservation and strengthens community stewardship. So without further ado, please join me in welcoming Dr. Enie Hensel.
Thank you, Thea, for that introduction. Can you hear me okay? And don't be afraid to interrupt me if my sound goes out. And I just want to make sure that you can, in fact, just see the presentation portion of my screen. I see a thumbs up, so that is fantastic. Okay. So today I'll be talking about how stingrays and scars shape seagrasses. I am very happy to be here, and thank you all for joining this afternoon. Today's talk is a modified presentation of two projects. The first one is an investigation during my time at VIMS, the Virginia Institute of Marine Science, exploring how stingrays who dig to find their food alter seagrass systems in coastal bays off the Chesapeake. And then my second is a portion of a project called Scar Maps. And for that portion, we explore the ecological impacts of boat propeller scars on seagrasses. And yes, I've embedded some of my stingray observations into that investigation. So if you know me, you know I like field ecology. I do think it helps to explore in nature to help find answers to today's problems we humans have created. This also means I will always need a willing village. And being back at UF at the Nature Coast Biological Station and within the Berry and Reynolds lab, I have found the most incredible team anyone can ask for, and I thank you. So now let's talk about some applied predator ecology. I hope your main takeaway from my presentation is that species interactions, including the disturbances they create, must be in and kept in the conversation for coastal management. Megafauna, or large body consumers, have shaped the ecology and evolution of ecosystems for over a millennium. But unlike terrestrial megafauna that have undergone mass extinctions, marine megafauna have survived and are still functionally relevant. Mega consumers, being long-lived and wide-ranging, regulate food webs, they modify habitats, and they alter nutrient dynamics through assimilation and transportation. Yet in today's world, the effects of these fauna are rapidly changing, where neutral or positive interactions are becoming, or at least are being perceived as negative and potentially detrimental. And the plot thickens. Because rapid global change has put about 48% of these animals at risk of complete extinction by the end of the next century. So before intensifying human activity drives waves of mass extinctions on marine megafauna, let's understand their role in structuring and maintaining coastal ecosystem function. But how can we manage and restore coastal habitats that have become vulnerable from shoreline development, intensifying extreme events from climate change, but are also critical areas for these large fauna? Well, Dr. Bea and her team are just a wonderful example to share. They were given the task to restore a vulnerable, degraded seagrass meadow that showed seagrass establishment success was only possible once they considered natural species interactions. And it was simple. They created barricades. They blocked grazers and bioturbators by poking some bamboo sticks into their restored grass. These sticks are biodegradable and temporary, so they can only stay until this meadow is large enough and can handle the ecological carrying capacity of large animals like sea turtles and rays. Her and her team's work is also a fabulous transition for me to suggest that large-bodied stingrays and seagrasses are a model system for quantifying the role of large consumers across levels of ecological organization. Why? Well, seagrasses are worldwide, and in tropical and subtropical seagrasses, you can find a diversity of benthic foraging rays. Sharing the conclusion of a recent review on ray ecology, there is still currently a need for studies at longer and larger spatial scales to measure their holistic effects on ecosystem dynamics. This need is because current studies are inconclusive to any general patterns. There's also extreme case studies that get a lot of attention. In the Chesapeake Bay, I can think of two right off the top of my head. One, J.J. Orth in the 1970s showed us that when you have a lot of rays with their feeding bioturbations, they can destroy an entire meadow. In the mid-2000s, a paper came out that said because large sharks are in decline, mesopredators are on the rise, which led to some miscommunication about rays being overpopulated. A media campaign came out stating, "Save the Bay, eat a ray," suggesting that by consuming cow-nosed rays, citizens would be aiding in the recovery of degraded Chesapeake Bay. Stingrays also have an economic layer to them as they interact with us and us to them, which I argue is further evidence to study their populations and their ecological role. Aquaculture businesses alter ray movement and feeding behaviors, and rays challenge fishermen daily like a deer to a soybean farm. Feeding and swimming with rays is a popular tourist attraction in the tropics, and that also changes rays' diet and movement behavior. As shown before, they can also affect habitat restoration, and they themselves are also a fishery. And it's one that's growing, be it from promotions like Save the Bay, Eat a Ray, or a more economical way to have scallops in restaurants. To study the effects of rays at larger spatial scales, my team and I conducted a 2-year experiment in a 100% restored eelgrass meadow located in the coastal bays of Virginia. These meadows are massive, like 9,000 hectares or acres. And it's said to be the largest successful restored seagrass system in the world. They also undergo very little human input or physical stress. So it enables me to isolate benthic foraging effects of these rays. These meadows are also a major migratory stop for very dense fevers of rays. And for decades, there's been a strong aquaculture effort to replenish these meadows with bay scallops. And they have recently rebounded so much, there are talks of when a fishery might open. So scallops are food that we and rays both have in common. And all these fantastic facts are a combination for an ideal study site to ask in pristine seagrass meadows what predicts ray foraging intensity, and then how do rays alter community structure as predators and habitat modifiers. For our study, we used a paired plot exclosure design. So plots were 15 by 15 meters, where exclosures blocked large organisms, including rays, and then the open plots allowed rays to come and go. Here's what those plots actually looked like. Additionally, to help answer ray foraging preference, our paired plots were along a scallop density gradient, and shown here with the heat map of density of scallops. We also conducted several snorkel surveys throughout that large meadow to test if seagrass composition affects ray foraging behavior. So if seagrass edge to bare sediment or any semi-patchy areas altered where these rays are foraging. And what we found was that ray pits were the most dense in patchy seagrass or along seagrass edges. And on average, each ray pit, creating a bare hole within that continuous grass, changed predator-prey interactions. So we're looking at the proportion of prey eaten, looking at scallops. Specifically, we found scallops were eaten more often in seagrass opposed to scallops along the edge or within those ray pits. And video evidence shows it is because reduced adult crab predation on scallops in the ray pits, which that makes sense to me. Crabs are not protected in the open pits like they are in seagrass, in the tall seagrass, and so they wouldn't take that risk to handle the scallop. And these findings also parallel some research in North Carolina that showing the exposed visibility from those ray pits also increased juvenile blue crab survival from the same thing, meaning big blue crabs didn't cannibalize. Comparing the total abundance and individual scallop sizes of our paired plots, open plots exposed to rays have significantly lower abundances of scallops compared to those exclosures that didn't allow the rays in, suggesting that, and this was also, this change in abundance was regardless of that localized scallop density, which is important to me because it suggests that rays are seeking out scallops for prey, even if they're hard to find. And then medium-sized scallops might be the most susceptible to ray predation effects. For rays, this target size also makes sense to me, being kind of like the best bang for your buck. It's not too small, getting all shell and no meat. And then they also aren't gape limited, meaning it can still fit in their mouth and they're able to crush it. And then leading back to the rumors of an open fishery in Virginia in the future, I'm curious how these effects might be implemented for bag limits and minimum scallop size. My main takeaways I hope that you have learned from sharing my study in Virginia, even in, okay, sorry, even in, I see that this is, okay. So even in these dense, luscious seagrasses, we still observe ray foraging preference to slight changes for seagrass patchiness and for seagrass meadow edges to bare sediment. And even in mild ray predation and habitat modification in relatively pristine meadows, we've had measurable effects of on faunal community dynamics. The Nature Coast of Florida is another example of a unique seagrass ecosystem known worldwide for being one of the largest, almost pristine systems. And guess what? Nature Coast also has rays, but it also has boats. And that is my poetic attempt of transitioning into the role of propeller scars as another disturbance in seagrasses. And don't worry, rays will be make a comeback before the end of this story. So as we dive in, I would like to remind you that seagrasses are important worldwide and they're critical habitats because of their structural complexity. And this is important at both the habitat and microhabitat scale. Think about a sea turtle within these meadows seeking refuge during cross-ecosystem migrations. Its canopy slows down currents, and that provides ideal nursery and feeding habitat. The structure of seagrasses does naturally come in all shapes and sizes. Even though I am focusing a lot on these luscious, continuous meadows, their beds can also be naturally patchy, having bare spots throughout the meadow, be it from detrital piles of aquatic plants, bioturbator pits, or grazed areas. Within the nature coast of Florida, we have, for the most part, very luscious seagrass beds. However, boat activity has been on the rise, and there's a growing concern in boat propeller scars and shallow seagrasses, particularly in those popular recreation areas. To address these concerns, No Restore has funded a co-production effort called Scar Maps, seagrass conservation through actionable research, having management areas for the prevention of scarring. One part of the Scar Maps project is to address two main ecosystem impacts questions. So, what are the consequences of scars on the valuable services that seagrasses provide? And then in severely scarred areas, is scar recovery too slow or not even possible without restoration? Assessment of these ecosystem impacts of scars just started this year, and we're collecting a wide range of data. Today, I will be sharing our first insights excuse me, into potential effects of scars on seagrass flora and fauna. To do this, we have 3 locations for our study. St. Martin's Keys, Weeki Wachee, and Anclote. We've chosen these locations because they differ in seagrass composition, as well as the fact that they have been identified as target areas from our end users and our managers. Within each of these locations, we then selected 8 to 10 sites across a scar intensity. Each site is 1 hectare or 100 meter by 100 meter, and then they're at least 500 meters from any other site. Within each site, we either took ecological measurements across the entire site or collected representative subsamples, as you can see with the X's here. So scar intensity, or density rather, was measured with our mapping team, and they measured the total linear meters of scars per hectare from aerial imagery. I then selected sites for each of our 3 locations across this gradient. So think, I have sites with really low, having almost no scars within that site, to medium to high, like this red one in this image, where it has over 1,500 linear meters of scars within a hectare. And the scale of our sampling efforts is dependent on how fauna and flora might be affected. So just taking seagrass composition as an example here, seagrass can be affected at both that microhabitat level, think on the left, like a scar being present or absent, thus removing the grass. We want to know what's happening right there at that local area. And that is where we collected data within these X's. But scarring intensity might also affect the entire meadow over time. Think about if loose sediment from multiple scars, maybe it reduces water quality and stresses bed growth. So by collecting these subsamples of these X's far away from any of these local disturbances, we're able to also measure how scar intensity affects overall bed composition. So in total, we have for our study 3 locations and 28 study sites in total. And to examine the potential scarring effects on the seagrass itself, for these measurements, as I just mentioned, at each site, we selected 3 unique scars and then collected pair data within the scar itself, the scar edge, and then seagrass that was at least 5 meters away from a scar or any other given disturbance. And we collected things like taking a biomass core, saying what plants were there and how many of them, what's that canopy height, and shoot density. To understand the loss of grass from just solely the scars themselves at the site or hectare scale, we just have a few estimates available to present today, but here on the x-axis, here on the y-axis, we have estimated biomass loss, and on the x, we have that scar density. And so our ranges that we are able to estimate thus far is that there may be a loss at the hectare scale from 100 to about 3,600 kilograms of seagrass biomass loss. 3,600 kilograms is almost 7,000 pounds. So that may sound like a lot of biomass loss, but when we think about this upper limit in the percentage of seagrass being lost per habitat, it's only about 1%. So there's some really luscious seagrass beds in the Nature Coast. Compiling a few metrics, we find scars are doing a few things to seagrass bed composition. So on the Y here, I have plant percent cover, and then on the X-axis, I have scar density, but I've blocked it into low, medium, and high, and then per location. So let's walk through this figure here together. As scar density increases, we observe, well, scars, them being themselves, they're creating patchiness, and this will increase with the number of scars. At both the bed and that microhabitat scale, we observe slight reductions in total plant cover. And as scar intensity increases, beds are becoming more seagrass dominated because of a reduction in the presence of structurally complex calcareous algae species. And that finding is a little concerning to me because I know that small invertebrates that live in the seagrass can be sensitive to the decreases in the number of these habitat-forming species. So for our seagrass-associated invertebrate sampling, we used covered cores in a paired design for that scar edge, and then seagrass far away from any disturbance to collect our seagrass-associated fauna. So far, we do have, thinking about just preliminary data right now, I can show you invertebrate counts. We do have lower abundances within scars compared to seagrass. But these data also show that if drifting macroalgae is present in the scar, invertebrates will also use that habitat. And I feel like this preliminary data also shows that the effect of a scar appears to be isolated to the scar itself without changes in habitat use, even on that seagrass that's right at the edge of the scar. There are still invertebrates present. To measure the potential scar density effects on fish communities, we conducted ARC fish trawls across all 28 of our sites. And since fish are so mobile, we're investigating scar density at both that hectare level, but then also scar density of giving a 100-meter buffer around each of our sites. And this will help answer if scar density within one of our locations is altering where fish are or that fish composition. So trawls were conducted in April, and today I'm going to focus on our most abundant species, which is pinfish. Trawl abundance efforts are on the left. Mean fish size is on the right. And we found no differences in the relative abundance or size of pinfish across scar density within each location. Reasonings for this? This might be due to pinfish being very mobile. And also Nature Coast, there's a lot of seagrass. So scars may not be having a strong enough effect for behavioral differences for a fish like a pinfish that's an omnivore. They are seeking small inverts, and as long as they're epiphytes on seagrass for them to slurp up, they're happy. So for next season, we are hoping to trawl a little bit later to investigate any potential fish community compositional differences. April might have been just a little too early for other fish species to have been observed in our trawl sampling efforts to have a measurable difference. I will note we're not specifically looking at how that microhabitat, scar, not scar, alters predator-prey interactions like I showed within the ray pits a little while ago. But you can look into the webinar's archives for more information. And Alex Wallace, a PhD student at the Nature Coast Biological Station and in Mark Hensel's lab, is leveraging our design to expand upon Teresa's investigations. So the last insight into scar effect on seagrass fauna that I'm able to cover today is exploring how fragmentation of scars may interact with natural disturbances, like grazing or bioturbation feeding pits from large bodied fauna, including sea turtles, manatees, and rays. As scars create edges, they can potentially facilitate foraging efficiency for these animals. Think of wanting a piece of pie and the ease of getting to the second piece of pie instead of you being the first one to carve out a slice. If scars attract these animals, like benthic foraging, benthic feeding rays, the combination could create non-additive synergistic effects in the loss or the recovery of seagrasses. And that's something I'm interested in investigating. These hypotheses sung really loud to me, particularly this past September when we were collecting data on the scar recovery rate portion of our study. And I can't go into methods today for our scar recovery, but basically just know that we were measuring and thinking a lot about what expands or widens scars, ultimately asking what can slow down their potential to recover. And so sharing some preliminary data on stingray feeding pits only. In April, we conducted 100-meter transect surveys counting ray pit density for all 28 of our sites. And then in late September, we conducted a paired transect counting the number of rays within a scar and then the number of ray pits directly adjacent to non-disturbed seagrass. For all 28 of our sites within April, we were seeing no effect on scar density to the number of ray pits. Just like same pattern that we saw for those pinfish relative abundances. But in late September, we did observe an average of double the number of ray pits within scars compared to its adjacent seagrass. This might suggest that there is a preference to that microhabitat change from the scars. And we'll be exploring these effects further with our scar recovery investigations in the upcoming years. So I hope you have learned that megafauna are still very much ecologically relevant in marine ecosystems through eating, some scaring, and modifying habitat. And I hope that you've learned that our actions can change their ecological role. And that species interactions and natural disturbances should be in and kept in the conversation for coastal management. And with that, I am happy to take questions and thank you again for listening.
Yay, awesome job, Enie.
Yeah.
All right, so as you guys can see, there is a poll there if you don't mind filling that out. But for now, does anybody have any questions for Enie? Feel free to type them in the chat or to turn your speaker on and ask her directly. I can start us off here. I do have some questions. So Enie, how can you tell the difference between a scar from other things like a ray pit or just it naturally being like bare sand?
I'm pretty conservative with defining what would be a ray pit. I will say this has been something I've been observing since the beginning of my PhD in about 2013 to try to, observe what is a ray pit, what is not a ray pit. And conservative meaning that if it's an old ray pit that could have just been macroalgae sitting there, I wouldn't call it a ray pit. Ray pits are pretty defined with the circular pattern, a little bit of this like rough removal of vegetation around the edges. and even kind of a really almost symmetrical depression towards the middle. And this is mostly for Hypanis species. For those that are interested, I'm not necessarily talking about cow nose for that symmetry. And for scars are really linear. And I have yet to see things naturally do this like really nice linear pattern. Um, Sea turtles can go in a linear pattern, but they often just graze the top like a lawnmower. Where here we're also really stringent on what we call a scar. And a propeller scar had to completely remove you had to see bare sediment. The grass had to be completely removed, not just a haircut form. Um, and I'm happy to expand on upon that if you need more.
No, awesome. That was great. Thank you so much. Um, Anybody else have questions for Enie? I have one in the chat here from Thomas Anderson. Uh, they said, thanks for your talk. I may have missed this, but how long do scars last?
You did not miss that. That is not something I went over. Scars, it's dependent on a lot of items, how far that scar went into the ground, if it was a haircut or went fully in. Also can depend on the type of species of the grass that it's within. Ranges could be from months to over a decade, or even 2 decades at some points, because it also combines with the natural ambient conditions, environmental conditions, like how much phosphorus or nitrogen is in the water and what are other stresses there. So a really ambiguous answer for you, and that is why these questions are being asked within Nature Coast, is because some of that is somewhat context dependent, and we're wanting to gather that. And we have it within 3 locations at different environmental conditions and different seagrass compositions. So hopefully within 2 to 3 years, I can answer that a little bit more specifically for you.
Awesome. Another one from Lori Morris. Great talk, Enie. Do you see migration or seasonal patterns in the rays in the Gulf area?
A really good question. I enjoy that one because there's definitely seasonal patterns within the Chesapeake Bay being more of a like temperate area. Um, as a scientist, so I'm born and raised in Florida, but as a scientist making observations, this is my second field season. So I don't quite have that minimum number 3 to make any set conclusions. I will say that this summer, I did notice that some more like active bioturbations occurred. I saw some within September where I was not seeing them so much in April. And so I think that does make sense. As water gets warmer, animals are going to move more, eat more, feed more. So there probably is some seasonal patterns, but again, I'm not going to put a black and white answer to that just yet.
Awesome.
I didn't notice any like migratory fevers, let's say that, which like Chesapeake is really known for that with cow nose rays and throughout the Nature Coast. I just did not observe that, and I was looking for it.
Yeah. Um, I have another question from Alexa Putillo. Great talk. Sorry if I missed, but how do you typically find the ray pits? Snorkel, or can you see them from drone footage?
You can see them from drone footage. I didn't do that. um, this was all via snorkel surveys, because it was either a set density within each of our 100 meter by 100 meter sites, or it was comparing seagrass adjacent to a scar. So it was like relative to the size of the scar, how many ray pits are within the scar, and then adjacent seagrass to itself. And so that's kind of the spatial scale that I was interested at. So totally possible to see them. I would say you probably need to ground truth them if you do that.
Cool. Um, I got another one from our good friend Amanda Chappell. Do you have any thoughts on the potential impacts of scars on blue carbon storage in seagrass meadows?
Hmm. Of course, you would ask that question, and that's a wonderful one. That is a portion of what we are doing further studies on, and we are taking core data and changes in sediment texture as well, in addition to what organics are within the system, within a scar, in seagrass on the edge, or just changes overall as scars get more intense or more dense. Um, that is just a collection, and we'll be like, we have not been able to process that yet. It's still in the works, and for next year, that's also something one of our priorities to make sure that we're collecting appropriately. So stay tuned.
Cool. Um, I do have one more question for you, and maybe we'll have some time for anybody else that needs to type in the chat. But um, has there been any change in attitude about stingrays being eaten? I noticed you said that in the beginning, and it was like a push to like for people to eat stingrays, and I just didn't know if that was still a thing or if it was changed.
Um, there has been a paper that came out from Dean Grubbs. He's at FSU that did in the scientific world explain, potential decline in sharks does not just correlation does not go to causation of an overpopulation of rays. And so in that regard, the conversation has quieted down a little bit. But rays are not typically thought of as very pleasantly when it comes to anyone trying to make money within the water, like aquaculture or different type of fishing. So um, slowed down in one regard, but still definitely a fishery and a nuisance animal, depending on um, what you're doing in the water.
Thank you. I have
Mhm.
another question in the chat from Mark Hensel. Does the relative importance of these small scale disturbances, turtles, rays, manatees, scars, change throughout the year? And does it change with latitude or dominant seagrass species?
Hmm. That's a really packed in question there.
Yeah, a lot in there.
Um, Change with latitude. Um, I would say yes, based on latitude tends to be changing your season duration, and so how often they can like affect an area is really shortened. Um, uh, and so in areas that don't have fast growing seagrass, or maybe really sparse, so like in North Carolina, where they have a lot of say haliduli, so shoal grass, maybe some rupia, really like really fine and somewhat sparse grasses. If they have a shorter season there, just because of literally the temperature of the water, that shortened potential growth season could really, ray pits could really affect that. Where then you go up a little bit north into the Chesapeake and you have really dominated seagrasses by eelgrass, which can can grow across 30 centimeters. On average, each seedling can spread 30 centimeters wide. So even though that is still a very short season, because of the dominant seagrass difference, those effects might not be as prominent as beds that are made out of more sparse or delicate grass such as rupia or haliduli. Um, and so that I guess that's what I'm thinking of with latitudinal changes, and then just how much time that disturbance can be like a press versus just a pulse disturbance of just being there real quick on a migration, or rays always being there. Scars, I do think tend to be seasonal because there are seasons when we're out and about having recreation in the summer or particular fisheries. And then in the nature coast, there's another piece where we get really shallow, low tides during the day. And so areas that would never be susceptible to your prop hitting the ground, you have to have that knowledge and know that in the wintertime, it's a lot more shallow. And so some of those areas might be more susceptible to scarring where they're not in the summer. So a little bit tangent of an answer for you.
No, that was great. You nailed all of it. So awesome. Any other last questions for Enie? All right. Well, thank you guys so much for coming, and please be sure to fill out the poll if you haven't already, and then we will see you guys back for next month's talk in December. So until then, take care, everybody.
Bye. And thank you guys again.